Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So I'm very excited to welcome Ingmar Riedl-Kruse, assistant professor at Stanford University. He runs the Riedl-Kruse Lab, and he will talk about interactive biotechnology and also about different platforms about, Biotic Games, and also how to create like low cost do-it-yourself kits with smartphones. Thank you very much.
So welcome, thank you for having me. So as the picture here indicates, one of the major themes in my lab that we're interested in is basically how can we make the microscopic world that we usually can see through a microscope actually interactive, right? We can look through a microscope, but we can't touch, we can't actuate, and this is the kind of thing we want to enable in very different ways and for many different reasons, as I will show you in my talk. So let's start with these two pictures, which I guess are kind of familiar to you, but just asking the audience, what's the difference between those? Say some different. One is tactile, okay.
One has an interface, or like a more accessible interface.
Okay, what else?
One probably has labels.
Okay.
One doesn't.
So
I think the users are younger.
different users, exactly. So essentially there are like 50 years difference between these two. In both cases we see computers and we see humans interacting with those. And as you appreciate, these early computers, they were primarily about number crunching. They weren't very effective by today's standards, and they were hard to operate. While those days, these devices basically provide a human interactive experience that is kind of child play and that is very, very powerful, right? And so 50 years of technological development made lots of things possible. And if we now look into biotechnology, we can actually see what's going on there right now, see similarities to what has been in electronics in like 50 years ago, and kind of try to project where should we go with that in the biotech space. And just to okay, so now the movies don't run. I don't know why. What you see here is are some microfluidic chips. Can we get the movies? I tested it before, it didn't run. Then it ran. Now it doesn't run again. Okay, let me try to walk you through. So what you see here is basically a microfluidic chip. It's like a network of little channels. It has valves in there, so you can move fluids in little packages around. It's used for diagnostics, for example. So if you have a droplet of blood and you want to do diagnostics of understanding which kind of disease markers or other things you have, you can imagine how these kind of particles are moved around on this chip and are kind of processed. And there are many relationships to these early computers. They can be run in a lab by expert people, but they're not readily available for day-to-day use, right? And so this technology gets better. There's something similar like Moore's laws to it. So can we basically get to a more human interactive experience with those ones as well? And so the first thing I want to tell you about is what we started with, is the idea of making games on these types of platforms, right? Even with the first computers, one of the first things people did was making games to kind of show how you can interact with those machines. So what are biotic games? We're interested in machines that have two basic features. One is it should really allow a human to interact with these biological processes at a small scale. And it should really have something to do with modern biotechnology, right? So what we don't mean is a pure emulation, and we also don't mean something more macroscopic like horse polo, which we could call ancient biotechnology, right? What you see here is a single-celled organism called Paramecia. It's relatively large, 250 microns. This is a very simple 2D microfluidic chip where you see these little speckles here. These are the cells. And then you see 4 electrodes. And if you apply an electric field, you can make those cells kind of swarm along the field lines to do a behavior called galvanotaxis. And so here you see kind of a joystick or kind of a gamepad where you actuate those electric fields. And then you can basically in real time on a computer screen see how these cells move. Now, if you put some virtual objects on top of it and integrate some tracking mechanism, you can play games with that, right? Like in the thing that you just saw, it was about getting points by swimming through these dots. In this case, you try to get kind of a virtual soccer ball into a goal, right? And so you could kind of envision and saying this is basically an interactive biology experiment, or you could say this is a game, right? And so in this sort of game engine, so to speak, you can now create many different games by just implementing different types of skins on top of it, right? Making different rules and things. Yes?
What is your goal with this game?
So the first goal, you mean what is the in-game goal, or what is the... So the initial research goal was like, I mean, could we make games on this type of platform, and even asking like, what would these games even look like, right? And before video games came around, no one knew what a video game was, right? And we have seen this tremendous evolution based on technology that led there. And so it's
Something about you can see how they move. Are you trying to teach people how these particular...
So yes, so there's lots of, so the initial thing is let's make games, let's make an interactive experience and show that it can be done. And then there's the secondary question, what is this good for, right? And I come to things like informal and formal biology education, right? So that may be one application there. So here see again kind of the setup. So it's very simple, right? This open chip, a hacked webcam, and then you basically project this on a screen. So this was kind of our very early first thing that we built and kind of laid the paradigm of what a game could be like on that sort of platform. And since then, we've basically gone quite a further down the road. And so the next system I want to tell you uses a different organism called Euglena. It's about 50 micrometer in size, a little bit smaller. It's green, it's photosynthetic, and it has this nice little red eye spot, which is actually uses to sense the 3D environment, and it has these flagella to kind of propel itself forward through the fluid. And so in this particular project, we wanted to set up a system where you can play through a smartphone, and where we also have a much more robust and reliable kind of game experience than what you saw in the previous one. So the educational motivations behind those is if you look at other kind of areas, especially mechatronics with the robotics and also computers with the national video game challenges, I'm always kind of envious because you see we have these beautiful toys that kind of engage kids and students to learn about these technologies, build something, have fun, and intrinsically learn. And we don't really have that to that extent yet in biology, right? And so here again, these cells that I just spoke about, these Euglena cells, right? Swimming through the fluid. A little bit about their biophysics, so to speak. So they have this eye spot, they have this long flagellum, and essentially they can roll, or they do roll kind of in a 3D helical motion through the fluid, thereby with their eye spot kind of sample 3D space. And then there's a feedback loop between the flagellum and this eye spot. And this allows them to basically, for example, go to the light if the light is very dim because they need light for photosynthesis, or go away from the light if the light is very strong because they don't want to get sunburn, right? And primarily here we use the negative phototaxis. And so here's on this setup, on this phone setup, the kind of games that we implemented is a very similar setup as before, but instead of electrodes, we have LEDs now, and light is much easier to control in many aspects, right? And so what you see a few differences to the games I showed you before. It's in color, right? So we made some advancement there. Which you may not appreciate is now these cells live for a couple of days in this chamber, so you almost get a plug-and-play experience, right? You turn the thing on, you can play, and then you can turn it off again. And we also already integrated a number of things that are kind of directly educational. So you see a scale bar here, you see the velocity down here of any sort of cell that basically is swimming through this object. Right now this cell is tracked, and then you see kind of the real-time velocity. You also see whatever cell is tracked, you kind of see the cell enlarged. And so the idea is, while you play with these things, there are many subtle ways how you can convey the learner or the player some things that is relevant about biology, right? And which you would not get if you would play a purely electronic version of that. So just to summarize again, so this is kind of this phone. You can actually completely build it yourself with a 3D printer and some simple electronics. And you can interact essentially in two different ways with these cells. One is you kind of tap on the screen with your finger and select a cell of interest. And the other, use a joystick to apply a light stimulus and and make the cells move. And as I will show you in the next few slides, there are kind of three modalities of learning or education that we have in here. One is you can build the system yourself, you can do inquiry by really doing experiments, or you can just play, right? So we tested this with a number of children on science fairs, and here you see kind of different age groups, what they kind of get out of it when they draw. And you see, for example, here this 8-year-old kind of drawing little legs on it, so which these cells don't have, but clearly reflecting that the child realizes this is something living, right? We also had some test questions about like how big are these cells or how fast are they swimming, which you could see from the game as I showed you before. And what we generally find is that above around 11 years, the kids fully get that. Below, kids enjoy it, but may not fully understand what's going on. And this actually also nicely aligns with kind of typical biology curricula in middle school when kind of most of the biology is taught. Here's basically the whole setup to build something like this. So you do some 3D printing. You have some very simple microfluidics, which you can make with some double-sided tape and some plastic. Very simple electronic circuit. And so idea is like build this, learn these concepts in an integrated way, right? And here's another one to do more serious experiments. So for example, instead of building the whole microscope, you can also put this interrogation chamber and the phone directly on a regular school microscope, which makes the whole thing cheaper. And then we made some apps where you can, for example, track individual cells under light stimulus or kind of measure a number of velocity traces, and you can ask hypotheses to the cells, for example, swim faster when the light is on versus not, right? And you can really do the whole scientific method on a system like that. And we also were kind of thinking, how could we integrate programming in there? So programming in Android, which we did, is a little bit hard, and so we recited to this Scratch children programming language, where we set up a simple model, which is actually all shown here. This is the whole equation that describes the motion, the animation of these cells. And then you can basically program games on top of that, or more serious experiments, right? And so we're now in the process kind of to engaging with teachers more directly and seeing how this could kind of flow in the school curricula or after school. And hopefully, in the long run, has a similar impact, like, for example, as Lego Mindstorms, right? So now let me go to a second project. Uses again these same euglena organisms. Yeah, okay.
Such a stick in the mud. Don't teachers ask you about more serious research questions you could be doing with these cells? Like, what are they good for? What do they do?
Yeah, so we actually...
Don't kids worry about that?
So I think the question of cells dying, maybe adults worry more than children. So we had a number of teachers actually coming to our lab and showed them and interviewed them what they found valuable and what not. So some of the things the teachers really found very valuable is already just having a screen where multiple people could look at it, or just pointing on a cell and tracing it. Some teachers were less excited about the game aspect. Some teachers feel games are very good for learning, others feel it's not there yet, right? So it really, now is the question how to really bring this to the classroom, right? And how to make it meaningful for the teacher. So it's really the next step.
Activity or something useful as opposed to speed. Like, could you teach them anything about what these cells actually do?
So, I mean, you can teach them, for example, they respond to light. I mean, already the concept that cells have an organelle that sees light, so to speak, right, and that can respond to it, and then always have a closed feedback loop that you always go with the light. I mean, that's an important concept, right? Another thing...
Part of me just wants to know more about these cells.
Okay.
Yeah, anyway, okay.
Good. So... So in the second project, we basically used the same cells, but tried a different set of stimuli. It's again light, but it's light from above, and so we have much more programmability, as you will see now. So this is a touch screen. What you see here are the cells swimming, and whatever you draw on the touch screen is directly projected onto these cells, right? And so you draw a circle in blue light, and so the cells avoid this blue light, and you can essentially trap a cell here. You can trap another cell there, right? So just to loop this, so watch out for this cell here, right? It's kind of swimming, and then it hits the light and it turns around, right? And so it's actually also a much more natural stimulus than what I showed you before, because here you really see the stimulus, while with the other one, with the joystick, some light from the side, it's maybe not always that directly recognizable, right? So what's the setup behind that? It's essentially a normal projector with a slightly different optics to make it small, and then you have a standard microscope with a camera to kind of feed it back into the touchscreen, and then you also have an eyepiece, right, a regular, so you can really also see not only the cells but also what you project. And here, what this movie is showing is actually you can do this on a slightly different length scale, right, by zooming out, and again you can see how the cells basically go away from this blue light and start assembling in these other areas. So this is what the machine looks like. This is the view through the eyepiece where you see the cells here, and you also see kind of what you draw. And this becomes important later, as you will see. So now you can do a number of things with it. For example, simple science activities. You can ask which colors do these cells respond to. So user can choose different colors, right? Like, for example, red or green. And noticing that the cells, whenever they encounter that sort of light, they don't respond. But if there's the blue light, they do respond, right? Very simple example of a scientific inquiry activity. And you can also make games on it. Here, for example, there's this apple, and you somehow need to steer the motion of the cells such that they get to the apple, right? Kind of try to make them bounce in certain directions. So we had this system then tested in multiple times in the San Jose Tech Museum. The setup is essentially like this big touch screen here, and then next to it, the setup, which we made semi-open so people could look inside, and also the eyepiece, right? And so we had people coming, and basically just we watching from the distance what people would do. And I want to walk you through a few examples. So what usually happens is people come to the touch screen, start drawing. It's a very natural thing. And after a while, they realize that there's something swimming in the bag, and that whatever is swimming there or moving is actually responding to the things that you draw. Here you see an example where a person within 2 minutes, so this whole thing is an image sequence of 2 minutes, where a person kind of initially trapped one cell and then made a little maze and tried to get the cell out of the maze, right? And so we can argue that we enabled a museum visitor to make a microfluidic circuit within 2 minutes without having ever done this before, and doing kind of a self-guided experiment. And what also very interesting, kind of having this eyepiece and the touch screen, if there were multiple people, usually only one person can operate one thing. So one person started looking through the eyepiece while the other one was drawing. And then, of course, they were recognizing that what one person is drawing, you see in the eyepiece, and then people started talking to each other and made kind of guessing games like, you draw, I guess what you do, and so forth. And which kind of really led to prolonged engagement on the setup, and people spending 3 minutes and more on there, which is really what you kind of want in a museum, right? And which again is something which we're really used to from physics, like hands-on physics and things in the active museums, but not that much in biology. I mean, apart from petting zoos, for example. And just to show you another example here, this is in collaboration with the Exploratorium, where we again use the exact same system, but now we use a Kinect as an input device, where the Kinect basically detects your body, right, and then projects that one into the microscope. And what this leads to is that, first of all, you kind of project the cells in your kind of own big space, right? You kind of, as a person, meet the cells there. But at the same time, if you look through the eyepiece, you see a little person dancing, right, inside the microscope. And that's actually something that especially children very much respond to, right? And so here you see kind of, right, like with this frame, how you can also kind of trap cells and move cells around. And so I think this really gets at a level where we could really have a two-way experience between a human and a microscopic organisms, both in our scale and in their scale. And we're kind of now developing, exploring, like, what are the best way to implement these kind of activities, and what does it really mean kind of philosophically for us, right? And the final thing I want to tell you in that sort of vein, what you've seen so far was always kind of single cells, but if you take like 1 million of these euglena cells and put them essentially in a closed petri dish, which is about this size, and then put light from below, you get so-called bioconvection patterns where the cells, whenever there's light, start swimming up. And then the cells accumulate at the top of the surface, but then the fluid gets denser, and then this dense fluid actually sinks down and drags the cells along with it, and then they swim up again. And you kind of get very dynamic patterns. And what you can do is, for example, if you have an initial, as this movie shows here, homogeneous distribution, you show a pattern on it. You see something like initially like a photograph almost, and then it develops further in more complex patterns, right? And so this is kind of a nice complementary system to the previous one, because here you kind of almost don't see the cells, but you really get a collective phenomena that involves millions of cells, right? But again, it's a phototactic behavior, right? So, okay, and this one is actually then also very easy to build, because it is just essentially this petri dish, and again, these cells kind of live in there for a couple of weeks. So, okay, so this was kind of our museum interactive explorations. And now I want to tell you something about how we can make biology experiments available over the web. And one of the main motivations of that is there are many students across the world, right, that would like to do biology experiments, and depending on how good your school is situated or how dangerous maybe even the organism is, there are lots of experiments you can't do. And the question is, could we facilitate students to work from any sort of electronic device over the web to do biology experiments? And we can, and we use again this paradigm that I showed you before with these 4 LEDs and basically looking at the phototactic behavior of these euglena cells. So this is essentially the website. It has one big live view of the camera, which is seeing your cells move. You have this joystick here. With this joystick, you control the direction of the light. And you have an external view, camera view onto the microscope. So here, if you watch closely, you see the different LEDs turn on and off, which is kind of important to give a remote user a sense of what they're actually doing, right? And if you follow closely, now, for example, the joystick points down here. So this LED is on, and then you see how the cells move downwards. If you look for a while, you also realize not all cells are the same. Some respond more, some less. The difference in sizes, right? And so generally, image data, video data is very rich. And so beyond this simple experiment like cells respond to light, there's much more to discover and to be analyzed in these types of movies, right? So this is a simple paradigm of kind of a cloud lab, an online lab. But it's very good for us because, again, these organisms are very stable and we can long-term culture them and really provide a platform that for the end user is almost as robust as a standard electronic one, right? And so in order to assess the utility for education, so I have a strong collaboration with Paulo Blickstein, who's a professor in education at Stanford. And so we went to San Francisco middle school, partnered up with one teacher who teaches multiple classes. And so we had on different days the number of students basically using our system, and then not only doing experiments, but also trying to analyze the data and then doing some modeling experience or some modeling activity around that. So that's essentially what the class looks like. It's pretty tough to fit in a 50-minute class, something where do experiment, analyze it, and model it, and then do pre- and post-test, which we also needed to do because we want to do research here, right? But essentially, that's the layout. So first about the experiments for the students. So there are different ways of how students can actually do experiments in a classroom. Either every student does an experiment on their own, or you kind of do a front classroom experiment where one student is basically doing it on the wall and all the students are kind of participating. What happened is that schools have firewalls, bandwidth limitations, all sorts of other things, which eventually led us to kind of gravitate more towards the model where there's one setup in the front of the class. But we would kind of envision that in the long run, when schools are kind of better suited for these kind of things, that really every student at the same time can run their experiments in parallel, right? So then the students, after this experiment, they would look at their data, basically like scrolling through the movie and kind of analyzing how many cells did respond to light, do they really align? We also made some kind of post-image processing where you're ready from a single image, you can recognize in which directions cells are swimming. And so, again, the main questions that centered around this was like, these cells are light responsive and they move away from light. And then we implemented a modeling environment. And so modeling is something that I think is still lagging in middle school compared to other science activities in my mind. And how does one even bring or enable a student to model these kind of complex cells? And what we decided to is building an emulation where we basically see this little box here, implemented the full 3D physics of this cell. And the student was tasked to kind of find a parameter set of 3 parameters where this cell would actually match the swimming trace here, and in purple, and this purple trace is due to a single cell that has been tracked. And the three parameters that the student had to align was the swimming speed, how fast it rolls around its own axis, and remember it has only one eye spot, right? So the rolling velocity really has an effect, and also how strongly it responds to light. And then students were basically kind of fiddling back and forth with these parameters and trying to get a good fit, right? In the future, we can think of many more sophisticated ways of how you really teach and engage children in modeling. What the post-test revealed is that, importantly, the children learned the content. And the other thing is that the students liked the activity, they felt agency, and felt that the performance was successful. And we also got very good feedback from the teacher as well, who said, like, this was one of the better classrooms, so children were much more alert. If you kind of now think of what are the pros and cons of normal hands-on biology experiments, so certainly the logistics are much easier, right? Like the teacher doesn't have to bring in all the microscopes and hassle and wrestle with the organisms. Of course, on the other hand, suddenly you lose a certain tactility that you have if you, as a child, kind of turn knobs in a microscope. And I think ultimately you need to kind of do both in a tandem, right? Certain things, it's really done well online, and you can really have the student focus on the experimental design and doesn't have to worry about hands-on skill. But in other cases, you really want to have that one as well, right? So, okay. So what's the long-term vision of that? How would that scale in the future? So what you see here is basically multiple of these microscope setups. So image is not very good, but essentially it's a camera here, the objective here, and then this little chamber. You may also see the tubes where we have basically the organisms in larger culture chambers, and where from time to time we basically flush fresh organisms into these chambers, and thereby having systems that can run semi-autonomously over multiple weeks, right? And so then we build out a cloud system where we have multiple of these machines, and then have corresponding user management where, I mean, how do you queue people? How do you give preferences, for example, for teacher over student, things like those. Also, if one of the machines is broken for whatever reason, right, how do you route people to a better machine? So with any sort of cloud system, you need to somehow maintain it and kind of monitor what's going on. We have that. But there's a special aspect here to the biology that you don't see in any normal electronic or computation cloud lab, and that's the organisms themselves, right, that may, for some reason, not respond as well, be dead, whatever. And so if you, for example, see this middle machine here, there are not that many cells. And so we basically implemented a framework where each of these machines, every couple of hours, runs a simple experiment, and then with object tracking kind of recognizes how many cells are there, are they responding and whatnot, and if the machine is kind of not properly working, people would not be routed to this one, and we would kind of get an alert to kind of fix it, right? So kind of really getting to this point where the end user kind of can log on and gets routed to something that gives a good experience. If we think about cost and scale up, so the whole technology behind it, as you saw in the talk before, is a very simple microscope, a very simple chip. You can run the whole machine with a webcam and Raspberry Pi, and if you put all this together, setup costs maybe $250. And if you then think you have many of those and have someone to maintain it, you can also think of that on average it costs maybe $250 per year to maintain a single machine. And an experiment essentially takes a minute. And if you plug in all these numbers, what you come up is that one of these machines, in principle, can run 500,000 experiments a year. This, of course, means like day and night, right? But people can access it from all over the world. And an individual experiment is less than a cent, right? So it's really something where you can think of, for example, addressing 5 million students in the US that every year go to a similar kind of grade level, basically really supplying certain biology experiments at scale and at low cost, right? And so that's something we're now thinking of how to really scale this up and make this happen. And of course, we're also worried, or not worried, I mean concerned about generality, right? So far, we always saw this light from the side. We can use other stimuli, as you saw from before, right? Like projecting from above. And we can also use different organisms. What you see here, for example, these are again Euglena cells. And what's coming in here is Volvox. It's also a microorganism, but actually a multi-celled ball. And if you shine light on this actually moves to the light while the euglena move away from the light. So you interact with a different organism. You also see, if you follow closely, how these cells kind of generate hydrodynamic flow fields that swarm other cells around. And so, again, lots of things to be discovered and to be seen. And one of the tricky things is always, if we want to bring these things stable online, is again the maintenance questions, like which types of organisms lend themselves well for being robust and don't need too much logistics on our end in terms of maintenance. Okay. So finally, I want to tell you about an earlier Cloud Lab project, which is an interesting contrast because it's working on a different length and time scale, and also is not as scalable as the first one, but on the other hand, maybe more have more utility for students actually building setups like those themselves. And the system involves basically petri dishes and an organism called Physarum that has a chemotaxis response. So focus on this one. So this is a petri dish about this size. In yellow, you see the organism, and in red, it's indicated where a robot basically puts food particles. The whole movie lasts over a day, so it's much slower experiment. And here an image is taken at about every 10 minutes. And what you can see is that this yellow organism essentially follows this trail of food, right? And you always see like it branches a little bit out, but essentially grows. Right here, initially it grows everywhere, then it finds the food, and then moves along the food trail. And the experiment essentially works like this, that the student logs in every couple of hours, programs this to the robot for a few more segments of the food trail, and then comes back a few hours later and see how the organism has responded. The system itself we built out of Lego, build a pipetting robot, and then have a flatbed scanner below that basically takes images of multiple of these dishes. So here's the interface where you can basically select your experiment, and you get routed to a particular one. You see your petri dish, and then you program essentially this food trail by, just drawing a line and then telling beforehand what's the time difference between those. And these are the robots that now execute these experiments. And what's interesting now is that, we have these robotics classes in school and after-school programs that now students could also build robots like that and then get kind of this whole mechatronics education more also into the life sciences, right? So, and then after the robot has executed it, the image is placed in the database, and you can come back and then see what you've done. So again, this is the robot. This is basically the pipetter, which is all made out of Lego except a normal syringe, so it's pretty low cost. And here in this, this is basically a standard server rack where we have 3 such machines, each of them having six of such dishes, which is important for us kind of just to try to understand the technology. If you have multiple machines in parallel, each with multiple experiments, how do you route and synchronize all of that? So that's actually what this kind of schematic is showing here. So you have multiple of these machines, each of them running multiple experiments. On the other hand, you have the user. And this setup is actually very different from the real-time euglena lab that you saw before, because before with the euglena, it is like one user checks out an experiment, owns it for like a minute and does the experiment and then gets put up again. While here, all these machines operate autonomously. The user only puts input into a database, and the machines query what they need to do and then do multiple things in parallel, right? So 2 very different modes of operandi. And what's interesting and important is that in the biotech space, we get more and more high throughput equipment, right, for pharma and research and so forth. And you can now imagine how you use this kind of high throughput, highly parallelized equipment to run many experiments in parallel, right? Rather, one scientist runs 10,000 experiments in one run. What about 10,000 students running one experiment each? And so I used this setup in a class at Stanford, a graduate level class that I taught, where the students essentially ran experiments over 10 weeks. And you see some examples where a student, for example, tests whether the organism responds more to, can distinguish between lots of food versus little food, how strong of a kink it can follow, things like those. And then the student would analyze the data, measure fractal dimension, growth speed, and then also make some models. And here's kind of another example of a already a little bit more simplified robot where you can, for example, program in 24 well plates, as you see here, food color gradients, right? Something that is very easy to do, is colorful. You can now think about more colorful mixes. And You also can do as more complicated experiments where you use bacteria that you induce certain gene expression that lead to colors, right? And so what this can really show is students how, give a feel for what industrial robots are actually doing and how robots can actually really facilitate experiments, right? Take the labor of the person and enable you to think more about programming and experimental design rather than physical labor and hands-on skill. And so kind of the vision where all of this leads to these kind of cloud labs, talked quite a bit about education, and you've heard about MOOCs and things like those. Another project that we're now starting is working with physicists, with a kind of a small select group of people that we give access to our cloud labs. And so they can run experiments based on their own choosing, but then really try to come up with interesting research questions and make models around that. Other more long-term vision is, of course, really using high throughput equipment on these kind of machines, on these kind of infrastructures, both for research and also education. And we can also think about citizen science, for example. So what if, so here, in case you don't know, for example, Folded and Eterna are projects where tens of thousands of people help kind of solve biological relevant problems. And so what if we kind of enable regular people to design their own experiments and thereby contribute to the scientific endeavor. So to kind of now step out a little bit conceptually, what I talked about is interactive biology, right? Like we want to enable people to do experiments in a very convenient way. And what do we need for that, right? We always need, of course, some biology, we need some sensor, and we need some actuator, right? And if you want to interface that with our standard electronic world, we would like to have a digital input and a digital output. Right? You can think about interactive biology where you don't have that, but as you saw, for example, if you want to build cloud labs or whatever, we eventually need to kind of interface with these kind of digital infrastructures that we are so heavily used to, right? And so if you kind of connect something to this digital realm, then you can make a close relationship or comparison to other chips that we know, kind of CPUs, GPUs, and so forth, right? Where you can say you have an input, and then something magically happens in some output, right? Some digital output. And the same thing happens here, except there's some biological process in the middle that is noisy, has some other properties, right? And so for that reason, we kind of call this sort of setup Biotic Processing Unit or BPU. And we can now also ask, like, what's a performance metric of such a BPU, of any sort of system, right? Like, you know how fast is your CPU clocked, all these kind of things, how many operations can it do? What would that mean for a system like that? And you could, for example, say, if you look back at this early system, we have this projector and this camera, you could say, let's assume we have a certain projector and a camera, HD, there's a certain amount of information processed through there, and use that as a measure, but that's not a good one, because if you have no biology in between, then all you get is some meaningless stuff out of it, right? So that can't be the answer, can't be a good measure. So we really need to take those into account. And the way we now think about it is really asking the question, if you as a user provide a stimulus, how much of a response do you see? If you provide a stimulus and don't see any response, you're essentially watching a movie, right? So really, what's the feedback that you get? And the way to capture this is actually mutual information, right? So from information theoretical perspective, basically from the response that you see, how much can you learn about your stimulus? And the more there is, presumably the more you can interact, or the more people can interact at the same time. And just to make this a little bit more concrete, so if you, for example, just have 2 LEDs, and it takes about 10 seconds for a cell to show a visible response, right? Then you can say 1 bit every 10 seconds, like within 10 seconds you can say which of the 2 lights has been on. If you have 4 LEDs, then you already have 2 bits. If you now think of this projector system where we can show from things from above, have multiple cells in certain sub-areas, you can actually get to higher levels. On the other extreme, if you think back about this Fusarum system, where we took only an image every 10 minutes, and the typical growth rate on that kind of length scale is in the order of 20 minutes, you actually get to a much lower bit rate, right? And if you now ask yourself, if you want to, for example, serve a certain number of children to do biology experiments, or maybe researchers to do certain experiments, and want to ask which system should you build, right? You could use these kind of quantitative measures of saying like, here's the kind of power of investigation that you have, right? Okay, so two final slides I want to talk about. So when we first had our games out there, some people asked like, is this actually ethical to play with living organisms, right? And I don't know whether the audience wants to respond to that in any sort of way.
Do the paramecium know they're playing game?
So,
Yeah, if they
They like the game.
so these cells certainly don't know because they are. Yeah, because they're not sentient, right? But what it prompted us is going to bioethicists at Stanford and really unwrapping this this issue, and basically coming up with a number of possible objections one could have. So there's of course the question of animal welfare, right? Like, I mean, do we hurt something? Do we cause pain? There's kind of a general question, respect for life, right? If you play with these organisms, this is something like you're disrespectful to single-celled organisms, whatever that it means. Actually, lots of these things we also got from online responses. So we have some YouTube videos where people are commenting, and so going through these, actually, those are some of the comments we find from there, can kind of characterizing. So some people are saying like, this is playing God, that's not what you should do. Some people say, this looks yucky, right? Like, some may say this is slippery slope, right? Maybe fine to do it with the single-celled organisms, but later you do it with mice, and that may not be okay, right? So don't even go there. Some people may say this is a trivial pursuit, right? Like, this is taxpayers' money. I mean, should we spend money on this? Couldn't you do something more useful with your time? Question about public safety, right? Like, is during this gameplay suddenly a killer virus generated that gets released in the public? There's also things about game ethics, right? Like, I mean, do we generate games that are good games, that are and joyful for the player, not frustrate, kind of, right? There are other questions which are kind of more longer fetched, but if you think about scientific discovery games, if things are discovered, who owns the discovery, things like that, right? So those were some of the major themes. Many of them have their relationship in kind of many other bioethical debates, right? And so what we then sat down and said, like, let's make a few recommendations, kind of setting some boundaries of what we want to do here to kind of really err on the safe side. So an obvious one is don't cause any pain or harm. So basically use non-sentient organisms. Always want to point out that these cells that you have, they are single-celled organisms. And when you put your pizza in your oven, you kill lots of yeast, right, which are kind of similar complexity. And you don't even think about that, right? We should certainly engage with the public and saying, why are we doing this? That's why I also give talks here, right? And saying, I mean, we have a mission to kind of engage people and teach, find new ways of teaching about biology and biotechnology, especially since it is an area that increasingly more influences our lives, right? We should show respect for life. It's always a good question, what does this exactly mean? But if you think about playing with your dog, right? I mean, you can throw a stick and the dog brings the stick back and you have kind of a positive relationship, or you could make a game where two dogs are fighting, which is something which we don't like, right? And so you can really think of how to incorporate that. And you should also have respect for the player, so really make games that in the end you think there's a positive outcome for that person, right? So those are kind of some recommendations, and so we published an ethics paper on that. And so I started my talk with some relationships between computer science and biotechnology. And here's a graph, so which kind of makes the point there's some sort of a 50-year gap, if you want to say so, between the two, right? There was kind of the early foundations of computer science, like quantum mechanics, can be maybe matched up with discovery of DNA structure. There's what we call the golden years, and then eventually there was the transistor and the first integrated circuits, which we now also have in biology, right? And you can basically fit a line through it and say 50 years, and we can, of course, also say when did the first video games come up and what did we do here? So we write on the slope with what we are doing. Right? Okay, so that ends my talk. So the main thing I want to highlight, what we're interested in is providing an interactive experience between a macroscopic human and a microscopic organism. We built now a number of these systems, learned a lot. There's a lot to be said about how do you make stable and robust technology. And then there are a number of applications. Lots of the applications right now focus on education, which I think is a rather straightforward one, because if you want to bring a million single experiments to a million students When the experiment is very simple, that's easier to achieve than bringing very complicated experiments to 10 specialized scientists, right? But these types of technologies in the end should kind of relate to each other. So that concludes my talk, and thank you for your attention. Yeah.
Can you comment more on the prospect of the citizen science aspect, like two prominent examples is like FODIT and also the astronomy, right?
Yeah.
And so, what do you envision doing?
So, so the general idea is like, could you not only have people involved in data analysis and problem solving, but maybe even in data collection and in experimental design? And so the Eterna project, which was on this one slide by Riju Das at Stanford, is already doing this to a significant extent, where people basically design RNA structures and they submit their design, and then these designs are synthesized and then tested of whether they fold properly. And so really getting people more and more involved in that part of the research as well. Another example would be, which we see a lot, that people with their cell phones, for example, go to the wild and take images of birds and other things, right? And basically help with the data collection. So.
I have also a question. What is the reason that you respond to different light frequencies? Is there...
So the euglena, why they respond most to blue, and so first of all, I mean, UV is the blue light is the more energy intensive, right? But it's also a little bit cheating, because if you really crank up the red light extremely high, and this, I mean, these projectors also have kind of some sort of a spectrum, right? They would also avoid that. So what we really chose to do something that gives a very clear, distinct experience to the user, where the blue gives a very strong response and the other ones do not. Again, it's a little bit, it's semi-cheating, because you should not mislead the user of saying like they don't respond to red light at all, right? So.
Does it anyhow affect them?
What do you mean affect them? So I mean, if you have very strong light for a long time, right? Like, I mean, you may get UV damage, for example, to the cell, right? May heat up. It really depends on what exactly is going on. But.
And are you thinking of like also then extending and like opening up to different stimuli that you go like beyond just like lights?
Yeah. So. So you saw the chemical stimulus with the robot, right? In the very first game that I showed, where we had like this electrical stimulus, right? So we tried to do more organisms, more stimuli, but the thing is that the light is very easy to handle stimulus, right? Because very easy to switch on and off. And the other thing is that these cells, these euglena cells, are now very easy to handle. And so we would really like to branch out, but the more complicated stimuli an organism is, the more complicated the technology gets, right? Yeah. Any more questions? Thanks again.
Thank you.